Hi, I'm Leslie Sturmer with the University of Florida IFAS Shellfish Extension Program. I, with other UF faculty, Andy Kane, Ed Flips, Eric Lovestrand, and Holden Harris, recently completed a study to begin to address oyster mortality in Florida's off-bottom aquaculture industry. There are certainly many challenges that this new industry must address, but this study addresses recent unexplained oyster mortalities experienced by growers in late spring and early summer, with reported mortalities at some lease locations of 50 to 90% of their oysters reaching market size. This is not just a local issue, but a problem facing growers in the Gulf of Mexico and in the southeastern United States. I'd like to first review these mortality events first seen at Alligator Harbor Aquaculture Use Zone in the summer of 2017 and again in the spring of 2019 and 2020, and there have been events in more recent years. Mortalities were observed in both diploids and triploid oysters, and I'd like to summarize these pathology reports. Samples were sent to the Virginia Institute of Marine Science as well as Florida Atlantic University. No major pathogens of concern, which could have been responsible for these mortality events, were not observed in either the tissues or shells, and that includes MSX and Dermo. And this provides further evidence that the Gulf of Mexico remains free of MSX, a major pathogen of concern for oysters in the Northeast and Mid-Atlantic states. Some other observations were the gill and digestive epithelial tissue were eroding, and that is certainly typical of dying oysters. Increased hemocytes. Hemocytes play an important or central role in the immune defense, and that's not surprising that these were increased as oysters were stressed. Edema or excessive fluid were found in connective tissue, again a stressor sign, as well as gonadal development in actually ripe males. And keep in mind that triploids can ripen, but they do not spawn. Other observations, mantle and gill abnormalities. The mantle basically is a tissue that provides skin or protection of the oyster's organs and plays another major function in producing the shell by extracting calcium carbonate from seawater. In addition, shells were sent to VIMS because of some discoloration of the shell that was observed. And it was noted that this is caused by extensive deposits of conchunclion, and that's an organic protein matrix in which the calcium carbonate is laid down. This is associated with colonization of the shell in which the oyster is trying to ward off something. And this coincides with rather large, almost extensive blooms of amphipods, very small shrimp-like organisms. These particular amphipods are tube producing, and they end up creating a lot of siltation on the surface of the oyster. All of these observations in the pathology reports fit the model for spring-summer mortality. Discussion concerning these mortality events began at a Florida oyster growers meeting held in January 2020. Possible causes of these events were certainly discussed, but there was no consensus reached as to what may be contributing to the events. All agreed that a systematic approach was needed to begin to understand these factors associated with mortality events. Then a few months later in May, a Gulf of Mexico oyster growers workshop was held virtually, in which experts from Louisiana State University all the way up to Virginia Institute of Marine Sciences presented what is known and not known in order to increase awareness of efforts, as well as to seek industry input about priorities that would be needed for future work. That workshop is available at the URL address seen below. Both the video and presentations are available to view. With some funding that became available in the same time frame through the University of Florida IFAS, we were able to initiate a preliminary study to determine basic but key relationships between production and health of cultured oysters and environmental factors. This occurred over 2020 through 2021. Objectives were threefold: monitor oyster production at 2 commercial lease areas, examine water quality parameters and phytoplankton abundance, and assess prevalence and severity of shell parasitism and Dermo disease. 4 oyster commercial farms were identified to be involved in this study. 2 were located at the Oyster Bay Aquaculture Use Zone in Wakulla County, and 2 additional farms located at the Alligator Harbor Aquaculture Use Zone in Franklin County. Oysters respond at the Auburn University Shellfish Lab in May of 2020. 2 triploid lines or stocks were produced, one from the traditional Louisiana tetraploid line, and the second from the new Florida West Coast specific tetraploid line. The maternal parents, or the diploids, were the same for both stocks, so these became half siblings. This allowed for the evaluation of performance of 2 genetic lines. The seed were shipped to Florida and then land-based nursed by UF and Cedar Key during June through September until they reached a size to be retained on a 12 millimeter screen size. Seed were distributed to growers in late September of that year, with about 1,000 seed of each line given to each grower. The average size for the Florida triploids was 21 millimeters in shell length, and then 19 millimeters for the Louisiana stock. Both weighed 1.4 grams in whole wet weight. These were deployed in 9 millimeter Vexar bags. Bags were supported by floats. 3 of the farmers used floating bags, and 1 farmer used the cages. The growers maintained the oysters during the culture period. Sampling was initiated in late January of 2021, 4 months post-deployment. Bi-monthly sampling was undertaken until the majority of oysters reached market size for this study that was 70 to 75 millimeters, basically a 3-inch oyster. So bi-monthly sampling occurred over 2 additional sample periods for Alligator Harbor, with harvest occurring in late May. There were a total of 3 additional sample periods for Oyster Bay, as the targeted market size was not reached until July 13th, and that was the harvest date then. Water temperature and salinity were continuously monitored at the lease areas, with the YSI SON maintained by the Department of Environmental Protection at Alligator Harbor, and then temperature and conductivity loggers placed inside an oyster bag at a farm at Oyster Bay. At each sample period, oysters were measured for shell height and whole wet weight, and live and dead oysters counted per replicate bag to determine mortality. In addition, oysters from each sample period were analyzed for health at the UF Aquatic Pathobiology Lab. Water samples were collected and preserved weekly during March through July, months when mortalities have been observed, with 2 sets analyzed monthly for phytoplankton abundance and quantity at the UF Aquatic Lab. Now let's look at some of the results. First, water temperatures. They were similar for both lease areas, declining throughout the first sample period and then increasing in the following sample periods. Seasonal differences certainly followed expected patterns. The grow-out period at Oyster Bay extended into June and July, with an average of 83.5 degrees Fahrenheit over that last sample period. At Alligator Harbor, salinity was high with very little variation, ranging from 28.3 to 30.7 parts per thousand over the sample periods. Lower salinities were observed at Oyster Bay, ranging from 23.1 to 17.8 parts per thousand over the sample periods. And this site is certainly more influenced by river discharge prevailing winds. So growth in terms of shell height, and that is the longest dimension of the shell. Many growers refer to that as shell length. That is displayed here with Alligator Harbor results on the left and Oyster Bay on the right. Orange bars represent the Florida stock and blue the Louisiana stock. At Alligator Harbor, oysters reached market size, again for the study, approximately 75 millimeters shell height in the second sample period after 6 months. but were allowed to continue to grow for another 2 months to observe performance under higher water temperatures, whereas it took an additional 3.4 months, or a total of 9.4 months, for oysters at Oyster Bay to reach market size. Greatest growth was observed at Alligator Harbor over the first sample period as temperatures declined, whereas greatest growth occurred over the third and fourth sample periods at at Oyster Bay. Although Florida triploids were larger than Louisiana triploids for each sample period and location, differences were not significant. Average whole wet weight of harvest-sized oysters at Alligator Harbor was 39 grams at 6 months, but doubled in weight after an additional 2 months. And again, these are the results for Alligator Harbor on the left side of the graph here. The greatest weight gain at Oyster Bay, displayed on the right-hand side here, was also observed over the period with warmest water temperatures as oysters reached harvest size. Values did differ significantly between the triploid lines during sample periods 3 and 4 at Oyster Bay, suggesting responses under more favorable environmental conditions, such as lower salinities, may be population dependent. In addition, here are some other measurements that were taken at harvest. Wet meat weight, as you can see here, was certainly higher at Oyster Bay during the summer months, but again, meat weight was higher for the Florida triploids as opposed to the Louisiana for both sites. We are displaying the fan ratio and cup ratios, but both of those measurements of shell indicate that Alligator Harbor and Oyster Bay meet the preferred sizes for the half-shell industry. In these graphs, you're looking at interval mortalities for each sample period and for each location. Mortality was negligible, less than 2% in sample periods 1 and 2 for both stocks and locations. However, over the next 2 months, mortalities at Alligator Harbor increased to 28% for the Florida stocks and 31% for the Louisiana stocks as temperatures increased. During this period, some growers at Alligator Harbor also reported high mortalities. At Oyster Bay, interval mortalities increased to 8 to 12% at the end of May, or sample period 3, which were 3.5 times lower than mortalities observed for Florida oysters at Alligator Harbor and 2.4 times lower for the Louisiana oysters at Alligator Harbor. Interval mortalities increased or doubled over the next sample period at 16% for the Florida triploids and 27% for the Louisiana triploids. Significant genetic stock differences were found during these last 2 sample periods at Oyster Bay. Phytoplankton is reported as biomass in these graphs, Alligator Harbor on the top, Oyster Bay at the bottom. The mean total biomass values were actually 4.5 times higher at Alligator Harbor than Oyster Bay. So dinoflagellate biomass, or peaks, which are colored in red, were dominated by 5 species, but the biomass values were below those associated with harmful algal bloom events in other ecosystems. Diatoms, which are shown in the yellow coloration there, they were the major species and contributed 61% of the biomass at Alligator Harbor and 39% at Oyster Bay. Diatoms are considered positive features in coastal food webs and certainly are preferred phytoplankton for bivalves. Other taxa were dominated by nanoplankton species, and that is in the green coloration in these graphs. Now let's look at health in terms of Polydora, or the mudworm. So we're looking at shell parasitism across locations and stocks. Prevalence, or observations of excavation, tubes, and/or blisters increased from 57% in the first sample period to 81% in the fourth sample period. Infection severity was relatively mild, that's the number of tubes observed per shell, with scores ranging from 0.4 to 0.9 throughout the study, and that's out of a scale of 0 to 5, so that's very low. and both these visual assessments increased with size and age of the oyster. Perkinsus marinus is a protista parasite that can cause Dermo disease in oysters. Weighted prevalence of Dermo was calculated from prevalence, which is whether hypnospore cells are present or not in the sample, and severity, which is the number of hypnospores that may be observed in a sample. This was scored on a scale of 0 to 5. Average values across locations and stocks were less than 1 for each sample period, which is considered to be a mild infection. Differences in stocks varied by sample period, but not significantly. So in order to look at all of this information, growth, survival, phytoplankton, environmental factors, and health, a statistical framework was used to examine how these affected the oyster responses in terms of growth and survival or mortality. First of all, the shell growth rates were significantly affected by the sample period or the culture period, lease location, and their interaction. At Alligator Harbor, the rate of growth was higher over the first 4 months than any other periods and at any sample period at Oyster Bay. During February through June, rates were higher at Oyster Bay compared to Alligator Harbor. Salinity influenced growth with no effect from temperature, health, or stocks. Looking at growth relationships in terms of whole wet weight, Alligator Harbor had higher weight growth rates compared to Oyster Bay, and rates at both sites increased significantly over the culture period, and that effect was 3 to 5 times greater than differences between the farm locations. Higher temperatures and higher salinities affected weight gain. No effects were observed or obtained from the health indices. Ploidy stock influenced weight gain at Oyster Bay, with Florida triploids having significantly higher rates than Louisiana triploids. To summarize the mortality relationships, these rates increased over time, duration of the culture period or sample period, and that was the strongest effect observed. Oyster Bay had lower mortality compared to Alligator Harbor over sample periods 2 and 3, with a significant interaction between farm location and the sample period. Higher salinities and higher water temperatures influenced mortality. Dermo was significantly higher in the last sample period, but did not demonstrate any patterns related to mortality, and there were no effects from Polydora on mortality. Florida triploids demonstrated lower mortality than Louisiana triploids, and these differences between the genetic stocks indicate potential to develop a line with higher resistance to environmental stressors and mortality events. I'd like to wrap this up with proposing or suggesting some management implications based on results of this study. Certainly difficult to do, given that this is a pilot study conducted only over one production cycle and with the limited number of commercial farms involved. But certainly one thing, a take-home message could be to be aware of stressful environmental conditions, certainly over the time frame when water temperatures are increasing over late spring, early summer. Would want to suggest that you might want to harvest your larger oysters prior to this time event and get them out of your production system. You may want to minimize handling stressors during this time frame, such as reducing the number of times you may flip your bags for aerial drying, as well as reduce the number of times you may tumble your oysters. Crowding is also a stressor, so you may want to reduce stocking densities of your larger oysters during this time frame. I also would suggest that you may want to evaluate both diploid and triploid stocks, see if one might perform better over this time frame. This study basically supports the present model for unexplainable mortalities, being that environmental stressors intersect with metabolic or physiological problems linked to oyster genetics and or ploidy to produce this mortality. I would like to thank all the growers who participated in this project, as well as thank you.